Okay guys, so this is chapter 2. We're going to be going over the eukaryotic cell. This should be review for all of you because Bio 3 did cover the eukaryotic cell and the organelles inside. So pay attention, there might be some detail that you didn't learn from Bio 3 or maybe have forgotten. Alright, so the outline begins with the microscope and cytology. So when you study cells, you want to use a microscope, and there are different kinds of microscopes. If you look at our little limits of our microscopes, this is a limit of the human eye, so our eyes without help can see paramecia, or we can see frog eggs. So you can see the unit measured here is in millimeters. When we go into very small structures like the cell, the unit that we measure cells in is micrometer, so it's 10 to the negative 6th of a meter. This is a very, very small unit, so remember that mu symbol here is for micrometer. And we can see that cells are measured in micrometers. So cells are very tiny. This is a standard epithelial cell, so this might be a skin cell. You have a lymphocyte here. You can see that lymphocytes are only 12 micrometers, whereas epithelial cells are 30, so it's smaller. We can see organelles like the mitochondria or the chloroplast, and we can see cilia, which are extensions off of the cell. So those are all possible with the light microscope. This is the kind of microscope that you'd see in class, but since we're not in class, you're going to have to use a simulation for the microscope. To see things that are smaller than that you can see with the light microscope, you use a sophisticated piece of equipment called an electron microscope. And so everything in this bracket here would be visible with an electron microscope. And now we're even smaller than the micrometer, we're in nanometers. So you can see nanometers is actually, just out of if you're curious, a nanometer is 10 to the negative 9th. So it's 1,000 times smaller than a micrometer. So let's go ahead and take a look at our light microscope. So you can see that from your outline, you will be asked questions on the light microscope. The light microscope, and then so you want to refer to your lab manual to get more detail on the light microscope, and there's exercises for you to label the parts and to know what the parts do. So basically you're looking at a light microscope here, and I'll just briefly go through the parts with you here. Sorry about that. So you have a microscope where you carry the microscope. In class, I would have you say, you would put one hand here and sort of cradle this portion. This is called the arm of the microscope. Sometimes there's a little depression in the back where you can hold on to it. The bottom of the microscope is called the base. Okay, and so then you would have a light source. So your light source is, you plug in the microscope. This is your light source. So the light microscope is called such because it does beam light through your specimen, and your specimen then has to be very, very thinly sliced, right? So if you look at number 1 under letter A of the first portion of your outline, it beams light through the sample, so the sample must be thin. About 2 millimeters of tissue will contain millions of cells. You have to get this so thin it's about 1 cell layer thick. If it's 2 or 3 more cell layers thick, it's too thick and the light won't pass through. Your cells, your samples will go on this portion here. So when you have a slide with a piece of tissue on it, you put it on the stage of the microscope. And there's a little clip you can see here. This is your stage clip that helps to clip your your slide in place. So what helps to beam the light into and through your specimen, so I'm going to put a little X here, your specimen would sit right there, is a piece of glass that you find in this portion here. So this is called the condenser. And the condenser is a piece of glass that helps to condense, kind of like if you think about a magnifying glass and how it focuses the light going through the magnifying glass. It's like that, but it does not magnify. The condenser just condenses the light, and the light's going to be focused on that letter X. So there's another piece of plastic, though, by the condenser that controls the light coming in, and that piece of plastic that helps to control the light coming in is called the diaphragm. And you can adjust this diaphragm to adjust the light coming into the condenser. Now, when you magnify, you're using 2 different lenses to magnify. the first lenses that magnify that the specimen light will go through is all of the different objective lenses here. So these are called objective lenses, and you'll work through this in your lab. Objective lenses have a range of magnification. You can see there's 4 of them here. 1, 2, 3, 4 different objective lenses. Each of them have a different magnification. They usually form 4x, then 10x, then 40x, then 100x. Okay, so those are called objectives. The objectives are on a piece of the microscope called the nose piece. And the nose piece helps to just hold the objectives, and you can switch, you can rotate that nose piece. So you can rotate this guy to change the objectives you're using, so you can go up in magnification. So your objective lenses, I'm going to star this because those magnify. Now, the second set of lenses that magnify are what you look through. Those are called your ocular lenses. And your ocular lenses always have a magnification of 10x. All right, so I'm going to also star this. So in your lab, so if you're looking at something where this letter X is, and you're using the 4x objective lens, the 4x objective lens is also going to go through a 10x ocular lens. You multiply the two magnification powers together to get your total magnification, which would be 40x. Okay. So the total magnification, how much you're actually increasing that image is going to be 40. So you always want to multiply the objective and the ocular to get your final magnification. Okay, so that was a very quick overview of the light microscope. And again, we'll go read the lab, do the exercise in the lab, and you'll be expected to label a diagram of a microscope and be expected to know briefly what the parts do and also what the how to achieve total magnification. I should also mention that in class, to focus your specimen, there are knobs here on the side. So there's a larger round knob here. It's a little bit difficult to see. It's a little dark here, but that's your bigger knob. And there's a second knob that's smaller that's attached. So those are the knobs to focus. The larger one is called the coarse focus or coarse adjustment knob. And then the smaller one is called the fine adjustment, fine focus knob. And these are going to move the stage up and down. And that closeness to the objective lenses is what's going to change the focus for you. So again, we'll be doing some virtual exercises with this. All right. So let's look at our other kind of microscope, which is an electron microscope. So if you wanted to see smaller structures, smaller than the cells, smaller than the cilia and bacteria, you want to use an electron microscope. So you can see this is quite different. This is a huge machine, right? So it's a very large piece of equipment here. And you don't use your eyes to visualize the object. The computer program picks it up, and so you will have to look at a computer image. So it's a computer-generated image. And there are 2 kinds of microscopes, 2 kinds of electron microscopes. So one is called transmission electron microscope. And this really requires very, very thin structures because the goal of this is to see inside the cell, so internal structures, with very, very high precision and clarity and at very small structures. So what you end up getting is a 2-dimensional image. And so this picture here is your TEM. So this is a cell. Let me outline the cell for you. So you have the upper border of the cell here, you have the division of the cell here, and here's another cell. I think there's a tiny cell there and then another cell here. What you see in this top part are the cilia. This picture is actually the same thing. So you can see the hair-like projections on this side. This is the same as the cilia here, except this has been thinly cut open. So if you remember from chapter 1, we talked about how a structure that is maybe a tube or a long cylinder like this, if it's bent, you can actually, if you cut through this portion, let's say, you might see a circle. But if you cut through a longer section of it, you'll see more of an oblong longitudinal cut. So those are this is typical. So cilia, even though it's all these long hair-like projections, depending on how you cut it, it'll look like little circles or it'll look like oblong ovals. In the second kind of microscope here, the scanning electron microscope, this one is meant to see surface anatomy very, very clear and very precisely. So it sweeps across the surface of the specimen, and it gives you a really wonderful three-dimensional image. So you can see, obviously in this picture on the right, how we have the cilia in 3D. So this is a nice look at all three of our microscopes, well, yes, that we talked about so far. So your textbook is full of images of this light microscope. Your cells are not pink. We know that because you're not a pink person walking around, but they do use a stain to visualize the structures within a cell. So the stain that's pink, typically they use the pink and purple stain. So a lot of the slides you'll be looking at are pink and purple. The pink is called eosin, that's the stain name, and the purple is hematoxylin. You don't really need to know those words, but those are the names of the stain. Then you can see the transmission electron microscope image, and you can see the scanning electron microscope image. So these are all images you'll see over and over again in your textbook. If we take a look at another example, right, so we can see these are two images of the same object. This is viruses budding from a cell, right? So viruses like the coronavirus would be emerging from your cells. And in this image, we can see the border of the cell here and all these dark circles. Those are the viral particles. And in this one here, we can see that we have a three-dimensional image of the same thing. So all of these little round units you see coming away from the cell, those are the viruses. Okay, so which one is TEM? Hopefully you identified the two-dimensional transmission electron microscope and the three-dimensional scanning electron microscope. This is just another quick image of three cells side by side. We have this labeled CW as a cell wall, so these are plant cells. We have M stands for mitochondria, the G stands for Golgi, the ER stands for endoplasmic reticulum, VAC is for vacuole. So hopefully this is all sounding very familiar to you. This is just an image to sort of appreciate the structures within a cell. And I want to show you some fun pictures using the scanning electron microscope with human tissue. So this is fat, so this is adipose. So you can see how adipocytes, the cells of fat, are these nice big round yellow cells. And then we have this sort of light tissue that this sort of spider web material, which is another kind of connective tissue that's helping to bind the fat together. So this is your adipose. And when you gain weight and lose weight, each individual cell either shrinks and gets smaller, so that's when you lose weight, or it can get enlarged and with more fat when you gain weight. This is the lung. So the lung is really very full of air pockets, and so it's a very spongy kind of tissue. All the black areas are spaces where air can pass through, and all the light blue are the very thin tissues of the lung. And so lung tissue is very delicate, and as we know with the coronavirus now and respiratory illnesses, that it can be damaged, and the damage can actually be long-lasting. This is spongy bone. So it turns out there's two kinds of bone. You have spongy and compact bone. This one, you can tell why it's called spongy because it does look like a sponge, so it has these spaces. So the bone itself is here, right? This is the solid portion of the bone. But in those spaces, in a living person, those spaces will be filled with marrow, right? Because we've heard that there's bone marrow, and that's where you find that marrow. Here's our skin. So this is a fun picture of skin. Skin has two layers to it. Many of you may already know this. So you have the epidermis and dermis. The epidermis is in pink, so this upper pink portion is the epidermis, and everything down from that is the dermis in green. Okay, so this is the dermis. They are two different distinct tissue types. The top is epithelial tissue, the dermis is connective tissue, but this is a way to appreciate how the top layer of skin is really flat and dead. And you can see the cells look like they're really ready to flake off your body. And that's what happens all the time, every day. You're constantly shedding your skin cells. You're losing skin cells all the time, because as those cells move up to the surface of your skin, they do die and they do leave your body. This is connective tissue. Connective tissue is there's very different kinds of connective tissue. So this is just one of them. This is called areolar. And don't worry about remembering this. This is chapter 3. We'll talk about areolar connective tissue. But you can see how sort of fibrous it looks. And then there are cells that are trapped within this tissue. Here's collagen. So collagen is a very big part of the connective tissues in your body. It's what makes your connective tissue strong. Collagen is known, maybe for most of us without a biology background, for I was going to say average person out there. You normally hear about collagen as like collagen implants, or, people in the healthcare industry, or the estheticians out there, wanting to plump up collagen, or trying to get your body to regenerate collagen, especially for the joints, but also for beauty, for skin. So this is what it looks like. It's basically kind of like a rope. So it's very strong. It has some stretch to it, but when that goes away, then that bulk that you have in your skin does fade. So you have the wrinkling that happens with age. This is human hair. So again, your hair is made of the same cells that your skin is made of. So you see how we have flat scales. These are dead cells that create the hair. They're just wrapped and coiled in a shaft. These are nerve fibers. It's like a fun way to look at nerves. They are basically kind of like your wires that help you charge your laptop and your phone. The outer part in gray, this is going to be connective tissue that protects the inner part, the purple, where the nerve fibers are. And then here we have another close-up look of nerve fibers. This is skeletal muscle. So this is the muscle that moves your body, and we can see that the muscle has these lines to it, right? So your skeletal muscle is a striped muscle, and you can see the structure here nicely in this three-dimensional form. All of the pink are mitochondria. So you see, you might have guessed that maybe these are blood cells, and that would be a good guess. But these are actually mitochondria. So your skeletal muscle is constantly using a lot of energy. Well, I shouldn't say constantly. When it's being used, it does use a lot of ATP. So it has a lot of mitochondria to help you generate ATP. And then I just have a nice picture of these cells. The cells come in different shapes. They have long processes. So you see how this is the main body of a cell. And that term for the main body, or the main part of the cell, is called the soma, S-O-M-A. And then any of these large extensions, like this extension that comes away from the soma, the general word is called a process. So cells have processes. This white blood cell also, you'd see these finger-like processes that come off the cell. And then this osteoblast, your bone cell, if you remember, or if you recognize osteo from maybe osteoporosis, right? This osteo means bone, and this light blue area is your bone. And they made this osteoblast, this cell, pink, and actually lives within a little cave, a little depression in your bone. Or actually not a depression, but it's actually a cave, right? Like a little opening in your bone. All right, so we're going to move on and look at the eukaryotic cell, but I'm going to do this in part 2, so we'll pause it right here.